This presentation will explain to you how the aldolase purification experiment will work. In our initial steps for aldolase purification, you're going to mince chicken breast and weigh out about 50 grams of your minced chicken breast and add homogenization buffer to it. Our homogenization buffer is a pH 7.3 phosphate buffer, along with a very small amount, 2 millimolar, of beta-mercaptoethanol. The purpose of beta-mercaptoethanol is to keep proteins in a slightly reducing environment, because once you break open the cells, you expose them to oxygen and an oxidizing atmosphere. And this can change the number of disulfide bonds in our protein. So we need to keep it with a small amount of beta-mercaptoethanol to prevent this from happening. The homogenization buffer also has 5 millimolar EDTA in it, just enough to chelate divalent cations and reduce protease activity. You're then going to homogenize it in the blender with short bursts, trying to avoid foam because foam is denatured protein. You're then going to carry out a differential centrifugation. You're going to spin out and pellet the whole cells that were not broken open by the buffer, all the connective tissue, making sure that is in the pellet. And in the supernatant, you will have the contents of the whole cells that were broken open by the blender. We're going to skip the DNA addition step to our supernatant solution. We have shown that our solution is not viscous enough through genomic DNA to need it, so we skip that step. We're then going to add ammonium sulfate after we put it through a mirror cloth and into a graduated cylinder so we can measure the volume. We're then going to do an ammonium sulfate fractionation of our protein. Now, proteins are kept in solution by interactions between the hydrophilic portions and the solvent. Now, remember, when you have a protein, the charged portions or the polar amino acids are on the outside of the protein, and those are able to interact with our aqueous solvent. By adding ammonium sulfate, we start neutralizing the surface charges, the positively charged ammonium reacting with the negative amino acids and vice versa for the sulfate. As you increase the concentration of ammonium sulfate, you reduce the chemical activity of the protein. At very high concentrations of ammonium sulfate, you're actually linearizing your protein, so you start to denature it as you add ammonium sulfate. And at higher concentrations, you are causing the protein to think it's more concentrated than it really is. You're diminishing the effective concentration of the water, and this causes the ammonium sulfate then causes the protein to precipitate out of solution. Now, there is no magic formula for how much ammonium sulfate it's going to take for any particular protein to precipitate out of solution. And that's because the concentration that's necessary to cause precipitation of a particular protein is relative to the number and distribution of charges and of the non-ionic polar groups on the surface of the protein. And then as the protein starts to denature, the number and distribution of the hydrophobic residues that are then exposed and rendered dominant as the charges are neutralized. So every protein has the amino acids in slightly different locations, and this can cause different amounts of ammonium sulfate being needed to precipitate out your protein. Even with a small amount of any type of salt, not only ammonium sulfate, but any type of salt, you do get a rapid increase in solubility of your protein. If you don't have any salt at all, we've previously learned that your proteins are going to aggregate and come out of solution. But assuming you have some salt at all, their solubility goes up very, very quickly. as you increase the amount of ammonium sulfate, maybe 10%, 20%, 30%, etc., different proteins will come out of solution at different rates, at different points in the amount of ammonium sulfate. So one protein might come crashing out very quickly. Another protein might have a more gradual decrease in its solubility. And the only way to determine for the protein that you're interested in what ammonium sulfate level is needed is to experimentally determine it. Is 10% ammonium sulfate, is it in the supernatant pellet? 20%, is it in the supernatant pellet? You go through and do that. So for our aldolase protein, we have previously determined that at 40% ammonium sulfate, our aldolase is still in solution, and other proteins have come out of solution, some other proteins. And our aldolase is still in solution with a bunch of other proteins also still in solution. At 60% of ammonium sulfate, now our aldolase will come out of solution because it has decreased solubility at 60%, along with a certain subset of other proteins. Remember, there's thousands of proteins in the cell. So by doing a 40 to 60% cut of ammonium sulfate, we're saying we're getting rid of a lot of other proteins that are not soluble between the 40 and 60% range, but you by no means are getting rid of all of the proteins except for aldolase in any way. So we're separating the proteins based on solubility, and we're using an ammonium sulfate table to find how many grams are going to be needed to achieve those specific concentrations. When you determine how many grams are needed, you want to avoid it slowly to avoid a locally high concentration. So you want to add your ammonium sulfate grams over approximately 5 minutes while stirring so that you can stir the ammonium sulfate in to get it into solution as quickly as possible. And then you simply do a centrifugation to spin out whatever proteins happen to have precipitated out at the specific concentration of ammonium sulfate that you are using. Then you want to get rid of the salt that's keeping your protein out of solution so that your protein can be soluble again. To do this, you need to dialyze to remove the salt. You want to make sure that your molecular weight cutoff is appropriate. Molecular weight cutoff is basically the measure of the holes in the dialysis tubing that you're going to be using, which will allow buffers and salts and maybe small proteins to leave the inside of the dialysis bag and go out into the solution. So you don't want to get your molecular weight cutoff too close to the size of your protein. If you had a 50 kD protein, 50,000 Dalton protein, you would want to use a molecular weight cutoff of 10 kD. So you don't want to use, for example, a 40 kD. That might be a little too close. You can buy dialysis tubing in all different molecular weight cutoffs, 1,000, 10,000, 30,000, 50,000. There's a variety of size ranges so that you can find something that's appropriate for your size of protein. And you put it into the dialysis bag and you put it into a solution, a buffer solution. Now, in addition to the salts leaving the bag, you can also use this as a way to do a buffer exchange. This is what we're going to be doing since we had it in a phosphate buffer. And for our next step, we cannot have a phosphate buffer or it would not bind to our column. So we're going to be using a tris buffer. So dialysis here is serving a dual purpose. It's allowing the ammonium sulfate to leave our dialysis bag, and it's allowing the phosphate buffer to exit and the tris buffer to enter with our proteins. You also want to do several changes of the buffer. the salts come to equilibrium, but there's still some salt in the dialysis bag. So you want to get rid of this buffer, put fresh buffer in, and allow even more salt to come out of the dialysis bag. So when you are finished with dialysis, your ammonium sulfate has left the dialysis bag. Then we're going to place it onto a phosphocellulose column. Now, a phosphocellulose column is generally an ion exchange column. The negative phosphates are going to bind positively charged proteins, and our protein will bind to the phosphocellulose column based on this charge attraction. But in addition to phosphocellulose being an ion exchange column, it's also going to be an affinity column for us, because the natural substrate of aldolase is fructose bisphosphate, has two phosphates on it. So the phosphates on the phosphocellulose column fit into the active site of our enzyme, and so the aldolase will stick to the phosphocellulose column because it's also acting as an affinity column. So it's acting as both an ion exchange and an affinity column. And we're binding it at pH 7.3. So at pH 7.3, we're binding proteins to our column. And then we're going to change the pH to pH 7.8 to wash off any proteins that would come off of the column based on this charge attraction. So we're making sure we're binding our protein based on two things. So our protein's binding very, very tightly to the column. Once our protein of interest, the aldolase, has bound to the column, then we can change the pH. So now it's only binding to the column as an affinity column and no longer binding based on ion exchange. Then we can specifically elute with our fructose 1,6-bisphosphate, which is the natural substrate and preferred even over a simple phosphate. So once we add the fructose 1,6-bisphosphate, this causes the aldolase to come away from the column, and we can then collect it in test tubes. We then are going to use a gel exclusion column to do gel filtration chromatography and determine the native molecular weight of our aldolase. So our aldolase is a fairly large protein. It's a multi-subunit protein, and we want to know what is the total size of our aldolase. So a gel exclusion column actually does not interact chemically with the proteins at all. Instead, there are spaces in all these beads, and these spaces have particular sizes, so small molecules are able to enter the beads, while large molecules are not able to enter the beads. So the path length for a small molecule might be in here, out here, in here, out here, in here, out here, in and out, and in and out. And so it's going to have a much longer path length than the large molecule, which is not able to enter any of the pores in the beads, and it's going to come down the column and go chunk, 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 chunk. So it has a much shorter path length than the small molecules do. So this means that we're able to separate by size on this column. And it's assuming that your protein is roughly spherical in shape. If it's oblong or cigar-shaped, part of the protein might be able to interact with the beads, and part of the protein might be able to enter the small spaces, causing your protein to run anomalously. But generally, if you have a roughly spherical protein, it's going to separate on the basis of molecular weight. And then the large proteins come out first, the medium-sized proteins come out second, and the smaller proteins come out third when you are collecting fractions in tubes. Now, when you start your column, you're going to have a certain volume of liquid in that column when you begin. Depending on the width of the column and the length of the column, you may have a greater volume in a column that's twice as long than you do in a shorter column. But you can compare your experimental results from one lab to another, even if people are using completely different sized columns. And that's because this amount of liquid that's in the column is known as the void volume. So a column that's twice as long is also going to have twice the amount of liquid that is in the column. So it's going to have twice the void volume. So when you are saying, well, how much volume does it take to elute my protein out? A column that's twice as long is going to have twice the elution volume as well. So what you do with any size column, whether you have a tall column or a short column, you calculate your elution volume divided by the void volume, your VE divided by your V0, and that's going to be a constant for any particular size of protein. So you can load multiple proteins at the same time on your column. So you can have a larger protein and a smaller protein together. The column's going to separate them out, and you're checking your fractions and saying no protein, no protein, no protein, and oh, here I'm reading my A280, and all of a sudden in these fractions now I'm getting a peak, and then it drops back down, and oh, now my 3 or 4 tubes later, now I'm getting a peak again. So what you want to do is you want to know how many fractions it took for your protein to come out, and you want to know the milliliters per fraction to be able to determine the total elution volume that it takes to get your protein of interest to come off the column. So you can have, for example, 2 proteins at one time coming off the column. The column separates them. They will have different elution volumes. These are the known sizes of proteins you're going to use on your column, and you're going to use blue dextran to determine the void volume of your column. Blue dextran is a very large molecule, and because it's so large, it's not able to interact with any of those pores in any of the places along the column. So it will come off the column before anything else will. So it's a good way to measure how much liquid total is in my column. So you add blue dextran to the top of the column, you say, when do I get my first blue drop out? And that is your void volume. Then you can add, for example, mixing beta amylase and cytochrome C, just as an example. And you can load both of them together on the column, and your column is able to separate them out because they are different sizes. And you can determine what is my elution volume for beta amylase, what is my elution volume for cytochrome C. And then you're going to make a graph. And you're going to say, well, if I know my elution volume and I know the void volume, VE divided by V0 for cytochrome C, you can plot that, and VE divided by V0 for carbonic anhydrase and bovine serum albumin, alcohol dehydrogenase, beta amylase, all the known sizes of proteins that you're going to put in there, and you graph the log of the molecular weight. So this is a semi-log graph. Or you could just simply take the log of the molecular weight and put it on regular graph on this axis versus VE V0. So then you have your standard curve for all your known sizes of proteins. Then you're going to run aldolase over your column. Now you don't know what's the molecular weight of aldolase. That's what you have to find out. But what you can determine is which fraction did my aldolase come out at? What is my elution volume for aldolase? And V0 is just for the column. So the elution volume for aldolase divided by the void volume for the column, that gives you that ratio for aldolase. So let's say it was 1.7. It's not going to be 1.7, so don't use this as, this is what she said it's going to be. This is simply an example. So let's say your VE-V0 for aldolase was 1.7. You come here, it hits the line, you go over and you say, okay, well, that's the molecular weight for that protein of interest that I have just run over the column. But it's not going to be that little because aldolase is a much, much larger protein than that. You are finding your native molecular weight, all the subunits together for aldolase, when you run it on the gel filtration type of column. This gel exclusion chromatography gives you the native molecular weight for all subunits. Then you want to find the concentration of your protein. As you go along your protocol, you're going to save samples. So when you finish doing differential centrifugation and you measure the volume of your initial supernatant in the very beginning, you're going to save a sample at that point. So you're going to call that your S1 sample, for example. And then after you finish your ammonium sulfate fractionation and do the dialysis, you can save another sample. Just a 1 ml sample is plenty. After you purify your protein on the phosphocellulose column, you're going to save a third sample. After you run your protein on the gel exclusion column, you're going to save your pooled fraction, all of your proteins together that were your aldolase coming off the column. You're going to save that S4. So you're going to have 4 samples that are going to be able to measure the progress that you made in being able to isolate aldolase. Because remember, protein purification is getting rid of the proteins that we don't want while keeping the proteins that we do want. As part of being able to measure this, you need to be able to say, how many proteins have I gotten rid of? How has my protein concentration dropped as I've potentially gone along this procedure? So to do this, you're making a standard curve with known amounts of a protein. In this case, you're going to be using BSA, bovine serum albumin. And you're going to be using known amounts, 10, 20, 30, 40, 50 micrograms of protein. And you're going to then put the Bradford assay mix on each of them and measure the absorbance. Now, I will caution you that your curve will not look like this. Your absorbances will not be this high. So please do not try to use this graph as something that you're going to use to determine your protein concentration. You have to do it for yourself, adding the known amounts of protein and measuring the absorbance of them and plotting them out on a graph. When you have done that with the known amounts of protein, then you're going to take each of your samples, your S1, and then your S2, and then your S3, etc., and determine the concentration of those. To do this, you would take a known number of microliters. So let's say we're working with S3, and we want to take 2 microliters of S3, and we're going to perform the Bradford assay on that, adding the Bradford mix, and then we're going to get an absorbance for those 2 microliters. Well, when you get an absorbance of 0.7, you come over here to the graph, you come down and you say, okay, let's say that's 24 micrograms. But we started with 2 microliters. So 24 micrograms divided by 2 microliters means that your concentration is 12 micrograms per microliter, or 24 divided by 2. You may take a different number of microliters, you may find a different number of micrograms, but in the end, you're going to be able to find the concentration of all 4 of your samples. In addition to the protein concentration, you're going to be doing specific activity assays on all of your fractions. And the basis for this specific activity assay is that aldolase cleaves the fructose bisphosphate into 2 3-carbon chains, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now, we don't have a way to measure this particular breakdown directly, but we can measure dihydroxyacetone phosphate reacting with NADH to form NAD. This is somewhat similar to what you did in the lactate dehydrogenase experiment, where you used NADH going to NAD, and you measured it at a wavelength of 340 because NADH has absorbance at 340 and NAD does not. So we can react dihydroxyacetone phosphate with our NADH to make glycerol 3-phosphate plus NAD. The enzyme that does this is alpha-glycerophosphate dehydrogenase. Now, please remember that when our aldolase cleaved the fructose 1,6-bisphosphate into those 2,3 carbon chains, triose phosphate isomerase then takes the glyceraldehyde 3-phosphate and also turns that into dihydroxyacetone phosphate. So for every one aldol cleavage, you get 2 DHAPs, and 2 DHAPs react with 2 NADHs to make 2 NADs. This is important because when you're calculating specific activity, you're going to measure how much your absorbance is falling per unit of time, very similar to what you did in that LDH experiment. Measure the dA/dt times the cuvette volume. What was the volume that you used in your, you had 1 ml of your specific activity assay buffer, and your specific activity assay buffer is going to consist of fructose bisphosphate, triose phosphate isomerase, NADH, alpha-glycerophosphate dehydrogenase, and you're going to be adding in this step right here. You're going to be adding in a certain amount of aldolase, which starts this reaction. And of course, there'll be a certain, amount of buffer in there. So you're going to be adding 1 ml of this solution, and then you're going to be adding a certain amount of aldolase. So remember that I said you had, in the protein concentration, you took 2 microliters of your aldolase. So let's say you just take 2 microliters of your aldolase again, so that your cuvette volume in this case is 1.002 microliters, 1 mil for the initial volume and the 0.002 for the 2 microliters. Or maybe you did 20 microliters of your aldolase solution. It can vary because you need to find a good dA/dt. You can't have too fast, you can't have too slow. So you might find yourself doing this multiple times to find a good dA/dt. You need to be able to get a certain number of readings. If you have it go, for example, from an absorbance of 1.8 to 0.2 in 30 seconds, it's too fast, and you'd have to use less protein. On the other hand, if you have it go from 1.8 to 1.6 in 3 minutes, it's going too slow. Use more protein. So you'll need to adjust the amount of aldolase you're using, and you do this by using greater or lesser volumes of your S1, your S2, your S3, or your S4, depending on what you are measuring in this specific activity assay. So you get a good dA/dt, multiply it by your cuvette volume, if let's say you use 20 microliters, 1.02 total cuvette volume, divided by 12.44. Now, if you remember, the extinction coefficient for NADH is 6.2. So why are we using 12.44? Well, in this case, the triose phosphate isomerase takes the G3P and converts it to DHAP. So we have 2 DHAPs using up 2 NADHs for every 1 aldol cleavage that's happening. So we need to multiply our extinction coefficient for NADH by 2. And here you put the milligrams of aldolase in that volume that you have chosen to use. And you know how many milligrams of aldolase because you did the Bradford assay to determine the concentration of your protein. So it's all coming together in your specific activity assay. Then you're going to fill out this fold purification table that's in your lab manual. And it's going to be filled out something like this. So let's say you have your S1, which is your crude fraction, and you got a dA/dt of 0.96 absorbance units per minute changing. And let's say it took 20 microliters of protein to be able to do that. Now, this is variable. I have 1.02 for all these, but this number will likely be different for you. I just did this to show you the math. So let's say it was in 20 microliters. So your total volume with your 1 ml cuvette volume of the specific assay mix itself plus 20 of the protein. And let's say you determined in that 20 microliters that you had 12 milligrams total of protein. You may have a lot less, you might have more, but this is just, again, a number to illustrate how to do the math. So you put it into that specific activity formula, 0.96 times 1.02 divided by 12.44 times 12, using each of these three numbers, and you get a specific activity of 0.0066. Your initial fold purification is always starting at 1. So then you do the same thing for your dialysis. And now your DADT is different and your total protein is different, but maybe you still magically got less protein by using 20 microliters still. You're putting it in that same formula, and now your specific activity is higher. You've increased your specific activity of protein. To determine what your fold purification of protein is, 0.0098, or the second specific activity, divided by where you started from, the 0.0066. So you've increased the purity of your protein 1.43 times. And then maybe after the PC column, you have a lower rate and maybe you have less protein. But remember that an increase in specific activity is getting rid of the proteins you don't want while keeping the ones you do. So your specific activity can go up even though your amount of protein is dropping. So you put that into the formula, you get a higher specific activity. That number divided by where you started from tells you your overall fold purification at the end of your S3. And you're going to do this for your S4. I didn't bother showing you S4 because now you understand how to do the math. So you can analyze single steps, a 1.43 increase here and a smaller increase here, or you can analyze, from the beginning, what is my total fold purification. So in your experiment, when you're being asked in your lab report to analyze the purification of your protein, you can discuss not only the individual steps, but the procedure as a whole. You also want to run an SDS gel on your protein samples. And an SDS gel uses sodium dodecyl sulfate, which has a charged end and a nonpolar end. And this is going to bind to your polypeptide and impart a net negative charge over the entire polypeptide. So even if you had a net positive charge to begin with, as our aldolase does, because it was binding to the phosphocellulose column, remember, by the time we're finished adding so much sodium dodecyl sulfate, it has a net negative charge on it, so it's going to move towards the positive electrode when we do an SDS gel on it. And having SDS completely coat your protein denatures your proteins and gives a constant charge to mass ratio. So basically, because it has that constant charge to mass ratio on it, it's based only on how your polyacrylamide gel is able to allow your proteins to go through. So the longer the chain of amino acids, the harder the time it's going to have getting through your polyacrylamide gel, so it's able to separate it out by molecular weight. The shorter proteins will be able to go through your gel faster. Now, it's important to note that your SDS gel is not breaking those disulfide bonds, and we're going to use a lot of beta-mercaptoethanol in here to break the disulfide bonds and allow the proteins to linearize. We added a small amount of beta-mercaptoethanol earlier just to keep our proteins in a slightly reducing atmosphere, but we're adding a large amount of beta-mercaptoethanol to allow any disulfide bonds to be broken. So after you've done that, you're going to load it onto a gel, and it's going to separate by size on the gel. different proteins with different arrangements may have, one or more than one band on them, even a pure protein. So if you had a pure protein with two different sized subunits, which are then separated by using beta-mercaptoethanol and using SDS to impart that negative charge on them, if you had a protein with two differently sized subunits, you're going to see two bands on your gel, even if your protein's completely pure. If you had a single subunit protein, whether that protein was a single subunit, or if you have a multi-subunit protein where all the subunits are exactly the same size, you're only going to see a single band on the gel because they're all the same size. So having more than one band on a gel does not necessarily mean that your protein is not pure. And in this case, we know that Aldolase is going to give us a single band on the gel because I'm just telling you straight up that all the subunits of Aldolase are exactly the same size. So you're going to run your samples on the acrylamide gel. On your S1, you're going to have a lot of proteins, fewer on your S2. After you've finished purifying it on the phosphocellulose column, maybe just one or two proteins, and then you can separate out by size so that you, at an S4, you have a single band of pure protein. And you're going to run these on an SDS gel next to a size marker of known sizes. So just in the same way you did for your gel exclusion column, you're going to have a variety of molecular weights, of known molecular weights that you run on your SDS gel in addition to your protein. And you're going to calculate for each of these known sizes, you're going to calculate the RF for your protein. And the RF is the distance that's migrated by the band from where it enters the resolving gel. This is a stacking gel that will be up here, and there will be a little line, and where it enters the resolving gel is where you will start measuring from. So the distance migrated from where it enters the resolving gel divided by the distance migrated by the dye front. So you'll have a dye at the very bottom of your gel. You'll run that dye to the very bottom of your gel and measure that distance from the resolving gel to your dye front. If you don't run your gel far enough, so maybe your dye front's only here, then you would just measure to here. So you need to make sure you know where your dye front is. So the distance migrated by the band divided by the distance migrated for the dye front, that's the RF for that particular size protein. And you're going to do this for all of your variety, different sizes of proteins. You will not have exactly these sizes of proteins on your gel. This is simply an example. So you're then going to make a graph of the log of the molecular weight versus your RF for each of your known sizes of proteins. Then you're going to determine the RF for your aldolase, and you're going to come to your graph and say, okay, well, here was my RF for aldolase. Come up to the line, come over. This is the log of molecular weight for my aldolase. And then if you take the inverse log, then you can say, this is the molecular weight for my aldolase. Now you need to say, well, I've isolated a single band on a gel, but how do we know that this is actually aldolase? And so what we're going to do on our gel is we're going to basically load everything twice. We're going to load S1, S2, S3, S4, and a marker lane. And then again, S1, S2, S3, S4, and a marker lane. And we're going to cut our gel in half equally, so each half has S1, S2, S3, S4, and marker. Half of the gel we're going to stain with Kumasi blue, which just stains every single protein. And that's going to look like that picture I just showed you in the last slide, showing up every single protein. The other half of the gel we're going to use to react with a specific antibody against our protein. So this procedure is called a Western transfer, where you transfer your proteins that are in your SDS gel onto PVDF membrane, and then you do an immunoblot reacting them with a specific antibody against the protein. So this Western transfer immunoblot protocol is generally called a Western blot, just to shorten the name. And you're going to visualize where your aldolase is by using antibodies. We're going to have a primary antibody. We're going to have a rabbit anti-aldolase. So this anti-aldolase antibody was made by injecting aldolase into a rabbit and allowing the rabbit to react against the antigen that is the aldolase and make IgGs, make antibodies against aldolase. And then you bleed the rabbit, separate out all the red blood cells, collect the serum, and the serum contains antibodies. Now, it will contain a lot of antibodies against aldolase, but it might also have antibodies against things that the rabbit has been exposed to that in the general environment. So it's called a polyclonal antibody that you've made, because it's not only a single antibody that's only going to react against aldolase, because there's potentially other antibodies in that same solution. You might get some cross-reactivity, not only against your aldolase, but against other antibodies reacting with other proteins that may be in your solution. But your darkest band will be your aldolase, because you're going to have a lot of anti-aldolase protein in there, because your rabbit immune system was stimulated multiple times by injecting aldolase into the rabbit. Basically, when you have a protein with a specific shape, all those different shapes are known as epitopes. So you can get even a bunch of different antibodies against all of the different partial shapes against the protein. We've said it's generally spherical, but there's an actual shape to it. So if you make a fist with your hand and you look at the front of your fist versus the back of your fist, you'll see that it might be roughly spherical, your fist, but it was different shapes. You have knuckles on one side, you have a thumb on the end, you have smoothness to the back of your hand. So all these are epitopes, specific shapes that an antibody can be raised against when you put an antigen into the immune system of an animal. So that's our primary antibody. Our secondary antibody is goat anti-rabbit. And in this case, a secondary antibody is necessary because there was no commercially available anti-chicken aldolase antibody made in rabbit. So because we don't have anything commercially available, there's no way to visualize the antibody. How you visualize the antibody is using a secondary antibody that binds to a primary antibody. So this secondary antibody is goat anti-rabbit, and it will react against any rabbit IgG antibody. So wherever our aldolase is, our primary antibody will bind. Wherever our primary antibody is binding, our secondary antibody will bind. And on that secondary antibody, there is an alkaline phosphatase tag. When you add a substrate, the alkaline phosphatase enzyme will cause it to turn purple wherever you originally had that aldolase on your PVDF membrane. So here's how it's going to look. The proteins are all transferred to a PVDF membrane. So something called a sandwich is made, where you put the PVDF membrane against your gel, you hold them together in a little holder, and then you run a current across it, causing all of the proteins to transfer to this very thin membrane and out of the polyacrylamide gel. And then you have a surface with all of the proteins that are now fixed to this PVDF membrane. Before you add the antibodies, it's actually important to add something called a blocker. And that's because antibodies are also proteins in addition to your aldolase. So any protein will bind to a PVDF membrane. Any protein. Since aldolase is a protein, it binds. Since the antibodies are proteins, they will bind unless you have blocked all the other available sites on the membrane with a very non-specific protein. So in this case, we're using bovine serum albumin. It's a very non-specific protein. Some labs use non-fat milk because milk has casein proteins in there. So it can be any type of a protein as long as it will not react with your antibodies. So for example, you would not be able to use chicken general proteins because the whole gamut of chicken general proteins will have some aldolase in there, so you'd be getting some cross reactivity between general chicken proteins that you would have added to theoretically block with the availability of aldolase in non-specific locations. So you add a protein that is designed not to react with your antibody. So once we assume that this entire PVDF membrane is coated with either aldolase and or other proteins that are binding to it, we can then bind the primary antibody to the aldolase, and then we bind the secondary antibody to the primary antibody. And remember, I just said that secondary antibody had an alkaline phosphatase tag on it that you can add the substrate to and get a particular color out of it. Now, alkaline phosphatase is not the only type of tag that is possible. There is horseradish peroxidase that causes a different color. There's chemiluminescent, so that when you add the substrate, it causes it to glow. Different companies have come up with different ways of showing you where that secondary antibody is. It just so happens that we are using an alkaline phosphatase system. So where do these antibodies come from in the first place? So when you, as I've said, inject your antigen, in this case the aldolase, into an animal, you inject it into the animal multiple times, and it's going to react with the B cells of your immune system. And the B cells then say, okay, we have detected something, some specific antigen, which is reacting with our B cells right here. And they go to your immune system and they say, make lots of whatever this is, and you have lots of different possibilities. In your spleen, in your bone marrow, you have thousands and thousands and thousands of different potential B cells that all have different specific ways to react with all the different antigens that you're going to be exposed to. Antigens against the pollen of trees, or if you're allergic to, animal fur, you're going to have antibodies circulating in your blood for all those. So you're upregulating basically the specific type of cells that are going to react with the antigen, and then you have all of these antibodies that your body is making, and those antibodies are then able to react with our antigen of choice. So the original cells have the antibodies on them that are then purified and floating around in your immune system to react with that antigen. So as I'd said, we then take serum from the animal's blood to be able to separate out all those antibodies that are floating in the serum. And again, you are going to have antibodies that not only react against your antigen of choice, but you're going to have other reacting antibodies as well. So they're called polyclonal antibodies when you get them from the serum. Sometimes, if a company has the money and a lot of potential market interest in a very specific antibody, it's a very expensive procedure to do this, but if they think that there's market share available, they will make a monoclonal antibody against one specific type of antigen. And so how you do a monoclonal antibody, and a monoclonal versus polyclonal, a monoclonal antibody means they're all exactly the same, and all of these antibodies are going to react against the exact same antigen. So you'd still inject your protein of interest into the mouse, and you'd still collect all the antibody forming cells from the mouse. then you're going to fuse the B cells, the antibody-forming cells from the mouse, with an immortal cell line culture, and fuse those together in something called a hybridoma. Now, the cells that are in your body have a limited lifespan. They will divide a certain number of times and then they will die. But what differs cancer cells and cells growing in tissue culture cell from cells that are in your and my body or the body of a mouse is that these cancer cells specifically are immortal. They do not die. So when you fuse these cancer cells that do not die with your antibody forming cells to make a hybridoma, now you have an immortal cell line that are all producing a specific type of antibody. Now, you're going to have lots of different hybridomas because you have lots of different antibody forming cells that you've collected from the mouse. So you're screening all those hybridomas, and this is the very, very expensive part here. You're screening to find which particular hybridoma, which particular cell mixture is going to react very specifically with my antigen. And this involves lots of tissue culture and lots of being able to lots of time, lots of money, lots of man hours to be able to do this. But in the end, you have a specific hybridoma, which is going to be an immortal cell line, which can then be grown in tissue culture forever. And the antibodies that it is making in a fermentation type system, not for bacteria, but a fermentation for tissue culture cells, basically. So it's fermentation type. It's grown in cell culture. So you can keep making these monoclonal antibodies forever. As long as you keep your tissue culture cell system going, you're going to keep being able to isolate monoclonal antibodies. So it's a lot more expensive to begin with, but it pays off at the end. So if you have a market need for a specific type of antibody, you can keep producing it in the end much cheaper, and you don't have to use nearly the number of animals that you would have to use if you were trying to do a polyclonal antibody. And it's generally much more specific because it's not going to have cross-reactivity. It's going to be only a single type of antibody reacting against a single antigen. So you're putting all of this together in your purification of aldolase, of being able to purify the protein. Explain to me how the specific activity in the Bradford assay allowed you to make judgments as to how efficient your purification was. Then you're going to be running it on the SDS gel and saying, here's all my proteins that are in S1, S2, S3, S4, and comparing that to the antibodies that are reacting specifically against aldolase. So of all those proteins you see in S1, for example, or the multiple proteins that you have still in S2, you can say out of all those proteins, here is where my aldolase was. So this is what you're going to be doing at the end of the experiment.